Hi, welcome to Mitosis. So this note is a bit of review if you remember a lot of this from sixth grade at Latin, but if you're new to Latin and maybe you've never learned this, this will be very informative for you. Even those of you who did learn it in sixth grade, I have a feeling you may have forgotten a few things. So this note will just review what you learned and possibly even introduce a few things that you didn't learn last year. Well, let's start. What is mitosis? First question on your note-taking guide. I just found this easy definition on Google, and mitosis is a type of cell division that results in two daughter cells, and they each have the same number and kind of chromosomes as the parent nucleus. Now, you can shorten that. You don't have to write that word for word. You can just say a type of cell division results in two identical daughter cells. And yeah, I don't know why they call them daughter cells and not son cells, but that's just what they call them. Usually this happens, it says here, ordinary tissue growth. So anytime we grow, anytime we're healing or repairing or replacing cells that have been damaged, we do this process called mitosis. And this leads to one of my favorite jokes of all time. Biology, the only science where multiplication and division mean the same thing. Because it happens that this is how our cells multiply. They divide. Now, before we can really talk about mitosis, you do need to know a lot about chromosomes. And as you see in this diagram, we could zoom in on a cell, and you'd have to zoom in into the nucleus of the cell where all the chromosomes are stored. And a chromosome is made up of all these strands of DNA bound and wound together very tightly. Here's the definition, official definition that you can put in your notes. A thread-like structure of nucleic acids and protein, you can abbreviate that to just say DNA. It's found in the nucleus of most living cells. And it has all your genes, all your traits inside the chromosomes, which we'll learn more about in our next notes on meiosis. Now, this is a diagram you're going to label in your note-taking guide, and I've kind of spelled it out here, and I put the numbers of each stage in the description. But this is how this DNA molecule, this long double helix, ends up looking like this X-shaped structure called a chromosome. It starts by, first, the DNA will wrap itself around these proteins that are specially designed to help the DNA to fold and coil up. It's kind of like wrapping a ball of yarn. Then, at this point, the DNA has already been an exact copy of itself. That happens previously. And when it's coiling up, it centers around this thing here represented by a red dot, and that is called a centromere. That ends up becoming the center of the chromosome. And what it does is it holds the two identical sets together end to end. But they don't stay in this shape for very long. As you can see, they start to twist up and coil up and become what are called sister chromatids. Again, I don't know why they don't call them brother chromatids. But the sister chromatids on each side of the centromere are identical to each other because they were once one strand of DNA that got copied. And then finally, in the fifth stage, they start to take on this shape that looks like this X. And with the centromere in the center, we now call this a chromosome. Each organism, each living thing has a unique number of chromosomes, and humans have 23 pairs of chromosomes, which makes a total of 46. And we'll talk more about this in our next unit, but I wanted to say this, not our next unit, our next note. I wanted to say something about that first because in a lot of these diagrams you'll see just a couple chromosomes. You'll see 2 or 3 or 4. Well, that's just to make the diagram easier to read, easy to look at. There are some organisms that only do have a couple or maybe a dozen chromosomes, but humans have 46 of them. So more about that later. Here are the phases of mitosis. You may remember prophase, metaphase, anaphase, and telophase. There's a mnemonic device that people use a lot. They just say PMAT, P-M-A-T. And then technically cytokinesis, I call it part of telophase. It's not a separate phase in itself, but sometimes you'll see it listed as PMATC. And there's also a phase called interphase that we'll start with. Excuse me. So as you can see in this diagram, mitosis is only one small part of the cell's life cycle. The majority of its time is spent in this phase called interphase. And interphase is where it's just doing normal cell stuff. It's making protein, it's converting glucose into ATP, it's just living its best life. But it's also at the same time getting ready for mitosis. It's making copies of the DNA. It's doubling some organelles because if in the end it's going to need a copy of everything before it splits in half. Otherwise, it will not be making two identical daughter cells. It would be making something else, some other type of mutation that we don't want. So the first phase technically is interphase, although you can also say it's the last phase after the previous set of mitosis. So in your note-taking guide, you'll see the diagram I've given you with all the circles. What I want you to do is draw, well, this one wouldn't have much to draw, but you can write on the right-hand side what's happening is normal cell stuff and getting ready for mitosis. If you wanted to draw anything, you can look at this picture and draw a couple things like the nucleus, the DNA on the inside of the cell, some other little squiggly lines all around it because it's just doing normal cell stuff right now. But now, prophase. This GIF is going to repeat over and over so you can watch it and use this to help you fill in the diagram. But what's happening on the inside is that the DNA, which is called chromatin at this point, this is when it is finally condensed into chromosomes. In this, you can see the chromosomes forming, those X shapes forming. And then they zoom in on it and show you that there's the centromere in the middle, the two chromatids on each side. That's the first thing that's happening, is that the DNA chromatin is turning into chromosomes. Then the membrane dissolves. You can see that it just dissolved here, so that the DNA and the chromosome can get out of that. That's where it's been this whole time. It's been protected inside the nucleus. And then these two organelles called centrioles move to opposite ends of the cell. Their job later on will be to separate the chromosomes. So I'll show you that again in the animation. Here's the centrioles moving to either end. The membrane just dissolved, and the chromosomes are the DNA turning into chromosomes. That's prophase. And metaphase is when the chromosomes line up in the center of the cell, all in a row on top of each other vertically. And this animation says something about motor proteins. There are actually little tiny proteins on each centromere that have like propellers on them. These proteins actually kind of move things around inside the cell and help them to get where they need to be. It's kind of fascinating that inside the cell all this stuff is happening. And you can also draw the spindles starting to form here. I don't have it written down, but that's an important step I forgot to type up. But the chromosomes lining up in the middle, you can draw that in your diagram, and then you can draw the spindles starting to grow on the ends of the centrioles, sorry. Then it's anaphase. In anaphase, the sister chromatids are being separated. They're being pulled apart from each other, and that's an easy way to remember it. Is at anaphase starts with A, and so does the word apart. The spindles are pulling them apart and splitting them in half, so that now we have the identical chromatids moving to opposite ends. As you know, that's going to be important later. So that's a good way to draw that. If you draw the spindles a little shorter and the X's being pulled in half and kind of looks like they're reaching out for each other, like no. So they're being separated now. Then there's telophase, and in telophase, The spindles are now dissolved. We don't need them anymore. Nuclear membranes reform around the chromosomes, and the cell starts to pinch down the middle. That's called a cleavage is forming. It's just basically a separation forming in the middle. And in the end, we have cytokinesis. Cytokinesis is the last part of telophase, where that pinch down the middle, the cleavage down the middle, becomes two separate cell membranes. And now we have two identical daughter cells. On your diagram, that's the result. You can label two identical daughter cells. And you can also label cytokinesis by just drawing an arrow at the cleavage part inside the diagram. Now, I thought it would be really cool to not just watch animations. This is a real cell going through mitosis. This is being observed under a microscope. Uh-oh. So let's watch that. So you can see those are the chromosomes in the center starting to move around. Those protein motor neurons are starting to move them around. There we go. It's getting ready for mitosis. Metaphase. They're lining up in the middle for metaphase. And getting ready for, there goes anaphase being pulled apart. And cytokinesis, telophase. Now we have two identical cells. Pretty cool. Now, there's one more page in your note-taking guide, so don't forget to move ahead. Is there such a thing as too much mitosis? The answer is a big yes, and that's what we call cancer. Normally, your genes control cell division and the cell cycle. It tells the cells when to do mitosis and when not to. And it really only needs to happen when you need to heal, like if you had an injury and you need to repair some tissue, repair some cells, or when you're going through growth, when you're going through any phase in life where you're getting taller, or even like things like fingernails growing and hair growing would be mitosis would be happening to make that happen. But when there's a mutation in a cell caused by any number of things can sometimes result in what we call cancer cells, and those are just cells that are multiplying too much, and eventually they start to clump together into a tumor, and in the wrong place. As you know, tumors can cause a lot of problems, but they also, because they're mutated, they look different. And you see in this diagram that doctors can look at under a microscope and sometimes can tell just by looking at it that these cells are cancerous. If you remember from our last unit on blood, when we looked at these blood smear from a leukemia patient, you notice that they had those white blood cells that called blasts. They were mutated, they were too big, they were misshapen, and after you had learned what it was supposed to look like, the blast, when it was mutated, really stood out, didn't it? So that's all we have for our review or our introduction to mitosis. And let me know if you have any questions. And move on to next topic will be meiosis, which is mitosis part 2.